Well, to start with, I would say good morning, buenos dias, to everybody here who came maybe all the way from Ecuador, I'm not sure. But anyway, hereby I open this academic ceremony in which Mr. Jaime Guevara Aguirre, I pronounce it completely not correct, I know, will defend his academic thesis entitled Diminished Insulin Secretion and Growth Hormone Counter-Regulation in the Etiology of Diabetes Type 2, mellitus, and Cancer, studies in 2 cohorts of human subjects with low insulin secretion and opposing metabolic phenotypes. It's a long title. May I invite you, Mr. Candidate, to present a summary of your study and the conclusions of your thesis in the next 15 minutes, and I'd like to give you the word.
Thank you very much, dear Prorector. Okay, I'm going to approach the diminished insulin secretion and growth hormone counter-regulation and the etiology of diabetes mellitus and cancer. And I'll do that by showing these cohorts, in which, as you can see here and here, they have diminished stature, as myself, but a little more. And here is with their families and with their families. Their disease is caused by growth hormone receptor deficiency, talking about proteins, and in here, excess of CDK cell proliferation inhibitor, P57, also known as KIP2, as you know. The basic thing about this is that they have different defects, produce the same short stature, but the case is that they have diminished insulin secretion. The first cohort is Laron syndrome also, and I will use interchangeably the terms. Laron syndrome is insensitivity to growth hormone due to a mutated growth hormone receptor, or that's what we call it, growth hormone receptor deficiency. So an autosomal recessive condition, the genetic abnormality is a splice site mutation in the growth hormone receptor gene, and the distinctive clinical features you already saw: severe short stature, diminished incidence of diabetes and cancer, improved brain health, and normal cardiovascular risk. The biochemical phenotype: elevated levels of growth hormone, improved brain and health, and normal cardiovascular risk. Diminished incidence of diabetes and cancer, I would like to reiterate. And the biochemical phenotype: elevated serums of growth hormone, low serums of IG1, BP3, and ALS that circulate as 150 kilodalton compound. The metabolic phenotype, besides the diminished insulin secretion, absent growth hormone counter-regulation, diminished insulin resistance. In this cartoon, you can see where the abnormality should look like. External domain of the growth hormone receptor, that's the mutation, inefficient signature transduction, therefore low levels of IG1, BP3, and ALS, that induces deficient growth. This deficient growth is because very little IGF access to the IG1 receptor. That produces also a very absent negative feedback on the growth hormone secretion and almost all absent signaling at the receptor, as you can see. Therefore, high levels of growth hormone, low levels of the 3 compounds. This is what it produced. This family, you have the father and mother, obligate heterozygotes, 3 affected children, and the rest normal. 14 years old, 19 years old, female, compared to 21, or this 9 years old compared to this 8-year-old sisters. This has been reviewed in several journals in the last couple of years. Besides the growth, we also notice that IG1 has to do with the harmony. And here you see the regression and the homozygotes and the heterozygotes and the wild type. And in here, that's the head circumference. And I can say that sometimes nature paints as beautiful images as the same greatest artists, like the Vitruvian Man. We published that in the JCM 2024. In morbidity, obesity, that's very, very important. That's the most important epidemiological risk factor for malignancy. And this obesity is associated with the malignancy. And what we found is less DNA breaks and efficient apoptosis associated with a low IGF-1. And over here, you have the Laron patient, the relatives, 1,606 and 99 Laron subjects, no cancer in the year 2011. This is going to change. But besides less DNA breaks, as proven by this COMET assays at 1 and 24 hours, you have efficient apoptosis, as documented here by two methods, including caspase positive cells and the cytotoxicity that reverses with the addition of IGF-1. But this is the most important finding, from my opinion. This is a simple to our OGTT, in which you can see that the GHRD controls very nicely glucose excursions. However, insulin secretion is very diminished as compared to the controls. And besides, let me show you other source of other proof of efficiency. Very low insulin levels here after a standard breakfast. This is 5 hours, samples every 15 minutes, and the GHRD control very nicely the glucose, and also they clear from the blood triglycerides. You can see here, and that's a very important action of insulin. And that partially explains the obesity as I can clarify later. This is the second cohort. This is an excess of CDK inhibitor, P57. We needed to compare, and in this case, the distinctive clinical features: severe short stature, intrauterine growth retardation, dramatic increase in early onset type 2 diabetes, and the metabolic phenotypes. They have diminished insulin secretion. You have 5 families, and with the white dot, you can see the affected people. See this father and his 2 daughters, 2 siblings. This family is very interesting, very affected by diabetes. In this family, there are 2 people that became blind after a few years of diabetes, and 2 have died with kidney insufficiency. That's why we decided to study 65 subjects comparing the 22 with Laron syndrome, 17 with the GRS, 26 adult controls that are relatives of the GRS, the 5-hour OGTTs, and 36, 11 with LS, 13 with the GRS, 12 adult controls. First, here's the panel in the 65. The LS, very low. This is the growth family. LS, very low, IGF-1, IGF-2, and BP3, as compared to GRS and the controls. Interestingly, in the adipocytokines, leptin, a little elevated, but not significant, but of the very nice protective adiponectin, total adiponectin, and high molecular weight adiponectin are relatively high. Here comes the most interesting part for me. You have here all the subjects at 36 and very little secretion of insulin. And with this little secretion of insulin, the Laron syndrome subjects, the GHRD, control glucose very nicely. That doesn't happen in the GRS, and that reflects the chemical phenotype, the biochemical phenotype 2, and the clinical phenotype 2. In this case, no one of the subjects, 6 GRS, 11 LS, and the controls, neither of them have been diagnosed ever with diabetes. And you can see this is the normal curves. Nevertheless, no diabetes. However, the GRS have still already diminished insulin. And this diminished insulin, what it produces, in my opinion, is early glycation of proteins. And we prove in this case that these subjects have early glycations of proteins, such as glycated hemoglobin or fructosamine. We studied the whole thing regarding, we're studying ghrelin, pancreatic polypeptide, every single thing. But the only thing that became positive, the finding, is that the GRS starts secreting the amylin, the insulin amyloid polypeptide, as you can see here. The significant. The LS are the ones that secrete the LS. Here's the GRS, and here's the controls. And I remind you that the amylin deposits in the pancreatic islets are a hallmark of type 2 diabetes, immediately suggesting why the subjects develop very early form of diabetes. This is published in the Diabetes Research Clinical Practice in the year 2023. So this cohort, what they have teach us here in this case, the GRS, here's the complicated pedigree, it's an autosomal dominant but an imprinted genes. And here's what the book says. This paradigm we challenge with the subjects, we suggest that they have low pancreatic reserve and inherited diminished insulin secretion, early protein glycation, initial insulin resistance, and they have this oversecretion of amylin, carbohydrate intolerance, further AIS, pancreatic failure, progressive hyperglycemia, established diabetes. And these old pictures. I'm very old, but I'm going to clarify that I didn't take this picture. It was taken in the 1900s. I did take the second one, this one. And here, in this old picture, you can see three people with Laron syndrome. The gentleman, this lady, here's father, mother, the uncles. And here you have Luis Germánico with his family, very, very nice person with an amazing memory. In summary, in the Laron syndrome, I could say that the absence growth hormone counter-regulation determines insulin efficiency. In the GRS, the growth hormone counter-regulation increases insulin resistance. So that's the difference. At the same level of insulin deficiency, they have different metabolic phenotypes, opposing metabolic phenotypes. So diminished insulin secretion and serum levels of both IGF-1 associated with less cancer and diabetes mellitus events, especially in the LS. Here's the team. Among the team, I put Professor Gavilanes here. And with that, I'll thank you very much, and I give the floor to dear Prorector.
Thank you very much for your nice presentation, well illustrated, and also gives a good overview over the work which you have done in the past, and which definitely will raise some further questions which we like to address to you. And we also have 2 minutes extra questioning time, which I'd like to thank you for. I would like to introduce several members over here now before our online audience who are present here. First of all, my name is Professor Steinbusch. I'm the pro-rector, and I am assisted today by Professor Driessen, who is our secretary. But more important, of course, is your promotion team, and there is one member available today, and that's Professor Dr. Gavilanes, who is sitting just opposite of me. We have, for your information, the audience, we have 6 members today in the opposition who are going to ask questions. 2 of them will be online. They will be on the screen coming over there. Okay. And the other ones, the 4 ones, are present here available. So without further ado, I would like to start the discussion. I hope you can hear us well. If not, let us know. And I would like to start with Professor van Engeland. She has an expertise in pathobiology of cancer at Maastricht University, and she also was the chair of your assessment committee, and I'd like to give her the word.
Thank you, Mr. Pro-Rector.
Excuse me, this is John. Excuse me. Yes. Mr. Director, we're having a very hard time hearing the proceedings. Okay. I could just barely hear it. I think both of us online are having a very difficult time of hearing anything other than
I will ask the technical team, Lorenzo in the technical team, Did you hear the questions from the online opponent that there are oh, he already doing. Yes, he is arranging it. I can see it over here. Thank you. Is it better now, Professor Kopczyk? Is it better now, Professor Kopczyk? Hello?
No, that's not enough. How about Professor Casido? Can you hear Professor Casido?
I don't know. We checked that. Professor Casido, maybe
Dr.
first of all,
Kopczyk.
can you hear us?
I cannot hear everybody at the auditorium well. There was always whispering from the tech support. I think that he was unmuted. So, and maybe we can improve the situation will be very appreciated.
Okay, we can see if the technical team, can you arrange this further with the both onliners? And can you put them also on the screen? I think we have to proceed.
Okay.
I'd like to give the word to Professor van Engeland.
Thank you, Mr. Prorector. Dear candidate, first of all, welcome to Maastricht. You have traveled a long way, all the way from Ecuador, to defend your thesis at Maastricht University. And I would like to start with congratulating you.
Thank you.
On this beautiful and interesting thesis that you have written under the guidance of Dr. Gavilanes and Professor Longo. For 30 years, you have studied growth hormone insensitivity in these cohorts. Okay, I hear an echo now. Yes. Oh, it's gone. Okay. You have discovered the largest cohort with Laron syndrome, and you were the first to describe the Guevara-Rosenblum syndrome. And some of these studies are described in your thesis. But before we can celebrate these achievements, we are going to discuss on these topics with you. My first question is about Laron syndrome. In chapter 13, you call these patients severely diseased, profoundly hurt, and I quote, the disorder will eventually be controlled and hopefully eliminated.
Can you please rephrase the last part?
Yes.
That's what I missed.
So at one chapter in your thesis, you write that you hope that the Laron syndrome will be eliminated eventually.
Yeah.
Yeah. So I would like to discuss that with you. One way to do that, let me first ask you, how do you think this disease will be eliminated?
This disease should be eliminated with genetic counseling. And it's a little sad to me to say that I put in the hands of the government in 1992, I said, here's the mutation. Please do and screen all the population. And then these people is very highly intelligent. They understand that if both normal parents, fiancé, whatever, the two potential parents, if they are going to have children, they might as well think it twice, because this is going to cost a lot if they have, in the 25% or so, if they have a child, it's going to cost a lot of money to the government and to themselves. So that's, I'm not talking about eliminating, but what I'm saying is that at least they should make a conscious decision. And this question maybe is the most important question I've ever heard in my life. And I am telling you that because that's what I'm telling since 1992 to the government, and they don't listen. They never listen to important questions. I do. And I appreciate very much your question. And we have learned a lot from the subjects. But the subjects suffer. This is a tragedy. I am a short person, but these guys are in this size.
Okay. So you advocate for early detection of the disease, right? And genetic counseling?
Yeah, once they are born, what I would like to see is that they receive the treatment knowing that the treatment never reaches to, I have one single patient I have treated to adult height, almost to adult height, but they have to receive permanent injections at a very high cost. Nobody can afford it in Ecuador. And now the government is giving the medications since 2 or 3 years. This is ridiculous. Just after 2, 3 years, these children are being treated. But it's not that simple, so.
Okay. So I don't know, but maybe you know that in Maastricht, we developed a technology called pre-implantation genetic testing. Do you know the technique?
No, I don't know the technique. I have heard something, but I can
Okay.
I can imagine what it is.
Okay.
I can imagine what it is.
So that is when IVF embryos are being tested for the disease. And then you can do embryo selection and only select healthy embryos without the mutation.
Wow.
Would that be an option for these patients?
I have never even thought about it, but that's marvelous. And of course, that'll be a beautiful option. That'll be an incredible option, actually, because that will solve so many things. This couple that want to have children, and they decided not to. That'll be perfect. That's a beautiful thing I didn't know.
Yeah, that question also comes with some ethical discussions, right?
Yeah, well, the ethical discussions in my country are going to be hard. That's part of, but I see the things this way. We physicians, we have to provide answers. The rest has to be decided by society.
Okay, thank you. Can I ask one more question? No? Okay. Thank you for your answer, and I'll give back the word to the prorector.
Thank you very much. Then the opposition will be continued by Professor Driessen. He is professor in pediatrics at our university, our hospital, and I'd like to give him also the word.
Thank you, Mr. Prorector. Mr. Candidate, I first of all want to congratulate you with your work, your very nice work. And personally, I haven't seen a thesis by a senior scientist with such a track record very often. It's quite impressive that you're still a PhD candidate and are such an expert in the field. And what I also appreciate that you studied rare diseases, and rare diseases can learn us a lot about the physiology and pathophysiology. So sort of experiments of nature. And I'm an immunologist, so I'm not an endocrinologist, so I don't know a lot about obesity and insulin, etc. But there's always something immunological to a disease. And I was intrigued by chapter 1 and chapter 7, that you wrote that there's an observation that Laron patients, they often die at young age because of common childhood diseases.
Yeah.
And that this vulnerability decreases when they are becoming older and when they're adolescents. Can you explain to me a little bit more about that phenomenon?
Oh, I would love to explain that. Mm-hmm. The only thing is I can't. The only thing I know is the facts. Mm-hmm. Somehow, until if it's even in these little towns, everybody knows when this child pass. I can give you potential. When they pass 7 years of age, they become stronger. And after initially they have a hair like mine, mm-hmm. the few little things that I still have, but the straight hair, and then become curly. But they die of common diseases of childhood. Mm-hmm. They die of pneumonia, gastroenteritis. They are very fragile initially, number one. Number two, they have a lot of hypoglycemia because they mm-hmm. highly efficient insulin induces this deep hypoglycemia that can take you to seizures and death. But the immunological part, this is something that I dream to do for quite a bit of time.
Did you do some research into this immunological?
No, that's what I wanted to do, but nobody. The problem is that You have to understand, I've been studying this almost by myself, using my father's funds, my family funds, some help.
Probably there's a possibility for collaboration then.
Immunological is something that is really very important, especially why this transition of somehow these children that are not strong, how do they become normal?
Yeah. Can I ask you, maybe with this in mind, when you have innate immunity, you have adaptive immunity, and when I was thinking about it, of course, when it's adaptive immunity, you build it up, and then maybe it can prevent the children at a later age. Did you consider? Do you know anything about immunoglobulin levels in these?
I would love to have.
Yeah.
You know, I was the first time I was going to study that with University of South Florida, and there was this famous immunologist out there, and I took the samples to them, and one of the doctors take the samples that I took, beautiful samples, and put it without noticing under UV light.
Yeah.
That was experiment. It cost me, but you know, what you're saying is it will be so nice to see several populations and understand, because everyone knows the fact. The fact is that they die before the age of 10, but after there, they are the healthier in the family.
And maybe it's a last small question. Yeah, looking at the mechanism of disease, that the growth hormone receptor is not functioning well, and downstream pathways are not signaling well. These pathways, they are also present in immune cells. You also put it in your thesis. Can you have some sort of a hypothesis that which part of the immune system will be most affected? You couldn't study it, but just
Well,
hypothesize about it.
Because you know what happens after the binding, the binding activations. You know, this binds to IGF-1 actually, even diminished, actually binds to several receptors, IGF-1 receptor, the insulin receptor, and the hybrids. So you don't know which way it goes, because at such low levels, you have more chance of the insulin being so efficient, the insulin accessing to the IGF-1 receptor via mTOR, for example. mTOR is such a beautiful protein, and I have some news to tell you. For example, when I treat patients with nutrition, just nutrition, trying to get mTOR by the way of the amino acids, that can activate it, and I have seen results in height. Why not in immunity?
Yeah. Thank you very much. I think mTOR is a very important player in the immune system.
Oh,
so I'm very happy about your theory that
mTOR,
it's.
to me, that's something very special.
Okay. Thank you very much for your answers. I'll give the word back to the pro-rector.
Thank you very much. The opposition now will be continued online, and we move to Professor Kopczyk. He's from the Department of Molecular Biology at Ohio University in Athens in the United States. And I would like to ask the technical team to bring Professor Kopczyk on the screen. And there he is. Welcome, Professor Kopczyk, and I'd like to give you the word to discuss with the candidate.
Yes, thank you. Can you hear me?
Yes, we can hear you.
Okay, thank you very much. And it's an honor to be here with you and with the candidate. As the candidate, and you know that it's very early here in the United States, so I hope I'm articulate enough to address some of these issues. Mr. Candidate, you mentioned that amylin secretion is greatly increased in your cohort of subjects versus Laron subjects. Could you further elaborate on that?
Hold on. Can you repeat the question?
He said that amylin secretion is more. Could you repeat it again?
Can you come back again?
Yes, can you hear me?
Team opponent, can you come back again with a question?
Yes, can you hear me?
We can hear you, but I will ask the technical team if they can make the speaker a little bit louder, because it's difficult to understand. Technical team,
Yes.
can you put it harder, louder? The signal, he's going to speak louder.
Testing, can you hear me? Testing?
Yes, it works fine.
It's working fine now?
Yes, it works fine.
Okay, Mr. Candidate, you mentioned in your presentation that amylin secretion is greatly increased in your cohort of subjects versus Laron syndrome individuals. Could you further talk about that more in terms of the mechanism? What do you think the mechanism is?
Yeah, this is very interesting. When you do a 5-hour OGTT, in these subjects, you administer glucose, obviously, and then very early they start secreting amylin, the insulin amyloid polypeptide, and that's completely new. You don't find that anywhere. But how do they do it? Because I believe that in the Laron syndrome subjects, they secrete less. But in the GRS, they secrete more, much more than the controls. And that's very important because amylin, anyways, has been documented since a long time ago that amylin deposits are basically the hallmark of type 2 diabetes. And that should be, I don't know if dear opponent, if you have ever looked at the pancreases of the Laron mice. I don't know that. But what happened in humans is that, what happened in humans in this cohort is that because they have normal growth hormone content regulation, is the low insulin level as opposed to the Larons, the low insulin level, what it does besides being low, is opposed by growth hormone that's fully active. Therefore, somehow they secrete more insulin amyloid polypeptide. And deposits of amyloid peptides are quite interesting, not only in the pancreas, you know that. So that is it's very good. By the way, the same manner that in one time in this conversation to you, I said that the high levels of growth hormone protect against infections against Trypanosoma cruzi. In this case, I'm going to suggest you something. Look it up at the mice with Laron, would be like the Ames dwarf mice that are somehow neurologically protected. So maybe there are some mechanisms why, because in both cases, insulin efficiency, both in the pancreas and the brain, may have some common pathways.
Yes, thank you. And this leads to my second comment at this stage. You mentioned the brain just now, the brain. You've concentrated a good part of this dissertation on obesity and insulin sensitivity. Could you comment on the comparison and contrast of cognition in Laron subjects versus the GRS subjects?
Yeah, we published that paper with Professor Longo, and we did a study. I took patients in that case, to his place in the United States, and we did several testing, cognitive testing. We did MRIs and everything, and the brain of the Laron subjects look younger. They have several areas in the cingulate area and the hippocampal region. I presume that even in the whole pathway, and I haven't, I would like to look at some places also at the hypothalamus. But we didn't look at that very nicely, but it will be good to see, for example, what's happening in the mammillary bodies. In all those places, there's an improvement, by the same fact that they have two things: low IGF-1, but don't forget, highly sensitive insulin. That, to me, that's more important. That's the point of the thesis. I'm more in the field. I believe more in a highly efficient insulin than in the low IGF-1, even though both are quite important.
Yes, thank you. For my last question at this point, by the way, that question, the last question about the brain was dedicated, referring to chapter 4, I believe, in your dissertation. So I'll need to go and reread that now with your comments. My final question is, we know that growth hormone corresponds to the growth hormone receptor, yes, but it also binds to the prolactin receptor. And in your both Laron subjects and the GRS subjects, could you comment your feelings about the involvement of prolactin action in the various physiological functions that you've described?
Prolactin could have some role, but never studied in the Ecuadorian cohort, at least. All the somatotropins share so many things. Maybe in cancer they have something to do. That'll be one thing, but I'm speculating. I'm a little concerned that this speculation can lead to something else. But maybe in cancer etiology, they can have some common ground. But that's the thing that I can mention.
Thank you.
Okay, thank you very much, Professor Kopczyk. Then we move to the second online opponent, which is Professor Cachedo. He was also a member of your assessment committee, and he is affiliated with the Department of Medical Biology at the San Francisco University of Quito in Ecuador, and I'd like to welcome him, and I'd like to give him the word.
Dear all, can you hear me?
Yes, we hear you very well.
Thank you very much, Rector, dear candidate. First of all, I would like to begin by congratulating the candidate. This document reflects a lifetime commitment to understanding two extraordinary human cohorts and the perseverance needed to follow them clinically, biochemically, and genetically over decades. Thank you very much, dear candidate, for this amazing thesis. And I would like to begin with my first question. How do you mechanistically reconcile that diminished insulin secretion is not deterministic, but context-dependent when modulated by GH-IGF axis disruption versus CDK and 1C dysregulation? And what does this imply for redefining type 2 diabetes etiology?
Well, basically, both, as I mentioned, both cohorts have diminished insulin secretion, right? That's the whole point. However, in the case of Laron, there's high levels of growth hormone, very high levels, but growth hormone is not active. Why? Because it cannot bind to the growth hormone receptor. In any case in the world, the growth hormone receptor, by definition, is mutated anywhere. Can be at the same level of the growth hormone receptor, can be in the JAKs and the STATs and the STAT5B, can be in ALS, several places. And this is one thing. But the metabolic part is that growth hormone cannot act, but cannot have any other effect, cannot have counter-regulatory effects. On the other hand, and the mutants with the CDK and 1C, because they stop the proliferation of some certain cells and G1 of the cell cycle, the cell cycle cannot proceed. Let me give you the example. In the opposite example, that's CDK1C that's KEAP2, that's the product, the protein. And in that specific case, you have the CDK1C and you have an excess of an inhibitor. That's a gain of function mutation. If you look at the Beckwith-Whitman syndrome, for example, that is a loss of function of the inhibitor, these patients are born big, number one, and they die of hypoglycemia. Why? Because they have a lot of proliferation of beta cells. So that satisfies you?
Yes, dear candidate. So now I would like to proceed with question number 2. If cancer protection in ELS derives from suppressed IGF, mTOR, RAS signaling versus metabolic energy restriction, low insulin, low substrate availability, which is more fundamental, and what experiment would distinguish them?
Yeah, well, regarding cancer and coming to this matter, the metabolic part of it, I agree with you. High energy, no matter what it comes, especially glucose, it's obviously glucose levels are by itself. For example, let me give you an example. Breast cancer. People with a blood glucose above 126 milligrams per deciliter, they develop breast cancer more frequently than people with a lower level of glucose. Why? Typical glucose enters the glucose 6-phosphate, the pyruvate kinase, then the cycle, then they enter into your lovely mitochondria, ATP, and et cetera. But the ratios, that's one part. Don't forget that every time that there is a cycle, after succinate, you create a GTP molecule. And that GTP is the main activator of RAS. The RAS and the MAP kinase pathway, the RAS protein, don't forget, it's present in 20% of the cancers in humans. So by itself, in one single example, I'm telling you that indeed, you are right, high energy levels are associated with that. That's one thing. But let me try to give you another thing. The cancer cells, because in the end, we are talking about cancer. Cancer cells love to get more GLUT4s implanted in the membrane. Why? They need glucose. When they soak glucose immediately, they even in the absence of oxygen, that'd be the Warburg effect. In the absence of oxygen, they can generate lactate, and the cancer cells need two things. Cancer cells need, number one, they need building blocks. They can get it from lactate, and they need energy. They can get it from lactate. Just that is the basic science that I know you love.
Thank you very much, dear candidate. Very compelling answers, and I have one last question for you.
Okay, dear opponent.
For the moment, how fragile is the protective phenotype in ELS, and what biomarkers would you monitor to detect its breakdown under the Western diet exposure?
Yeah, that's what I'm afraid, that all this protection that we are seeing in the LS is going to vanish. It's going to vanish by the Western diet. When I discovered this population quite a long time, these guys used to have a very traditional diet, but the roads came, and with the roads came the Western civilization. Now, hamburgers and pizzas and especially ultra-processed foods, these guys are becoming just too big, number one. That's number one. Number two, in the same area, what they have is a lot of contaminants coming from the illegal mining. So they have two things. So no matter how much protection you have, and my impression is I think when I completed these studies, if I live by the year 2027, I'm going to see an increase in both diabetes and cancer, because you know, in Ecuador, cancer in the southern provinces is rising up in a crazy manner, and so there's no way that they can escape, no matter how protected they are, genetically speaking. Thank you.
Thank you very much,
Thank you,
dear
highly
candidate.
esteemed colleague.
Thank you very much. Then the opposition will be continued by Professor Gazzolo. Professor Gazzolo is involved as chair of the Department of Neonatology at Chieti University in Italy, and I also like to give him the opportunity to discuss with you.
Thank you, Mr. Prorector. First of all, I want to congratulate the candidate with the promoter team for this brilliant dissertation that opened me a series of questions that I would like to elucidate with you. I want to combine the two with my question. First, the main two chapters of your thesis, and can you explain or to elucidate whether these two cohorts that you study have diminished insulin secretion and conversely have an opposed metabolic phenotype?
The opposing metabolic phenotype because of the diminished insulin secretion. All right. That's basically the thing. Once, first I discovered that 1/3 of the world's population with this Laron syndrome. And then I said, gee, this is diminished. I don't know what it means, really. I know that they is highly efficient, but how? And then by coincidence, this guy's walking. This guy he was sent to me by an endocrinologist, and she said, he has some strange type of diabetes. When I look at them, I say, gee, the Barkey hypothesis is working, And this is fascinating because this is something that I'm asking Professor Gavilanes to help me out with, because this is in the future. I'm talking about some studies. The fascinating thing that the Laron patients are born almost of normal size, and then very soon after birth, their SDS declines, and so they have normal body size, and then they go down immediately. But in the case of the CDKN1C, they have intrauterine growth retardation. They are born very, very small, and then they grow, well, relatively normal, but they still are very short. But the metabolic phenotype that you are asking, I have always thought that maybe these people have some kind of early glycation even in utero. Why? What I think is because the children of diabetic mothers, how are they born? They're big. Why? They have plenty of glucose. They have excess glucose. And maybe that excess glucose turns on the insulin secretion in the fetus. Maybe over here, no matter how much glucose the mother has, it's going to be a little bigger, but it's going to be short anyways. That's the IUGR. So that's the part of the initiation of the opposing metabolic phenotypes. And that expresses in adulthood. In adulthood, what you have is basically two groups with diminished insulin secretion. But in one case, you have the growth hormone opposing to the action. Growth hormone is one of the counter-regulatory hormones. So you have, in the case of the GRS, they have normal growth hormone, not only normal growth hormone, they have normal responses. If you inject them with growth hormone, they respond with IGF-1, BP3, and ALS. Very strong, very robust responses. In the case of Laron, they can have tons of growth hormone. They don't have a receptor, therefore they don't have any growth hormone action. And your question is also very important because many people assume that it is growth hormone responsible for the counter-regulation. It is IGF-1. I think you are correct in that sense.
Thank you. May I move to the second question? And I was very curious to better understand why the cases with Laron syndrome became obese.
Oh, that is something that they have very low insulin. They have very low insulin. By the way, besides that, this is very important to notice that in the two cohorts with diminished insulin secretion, they don't develop metabolic syndrome. That's very important. But now, could you please focus a little bit more so that just the obesity part of it?
Yeah.
Okay. What happened is that this highly efficient insulin, the only thing it does is gets triglycerides and everything inside the adipocytes and locks them in because it's highly efficient. And the other case is diminished but ineffective. And that justifies the gross obesity in one case and a little overweight in the other case at best. But obesity is justified, I think, just because of efficient insulin without growth hormone opposition.
Thank you. My last question is a prospective question. If your findings can be useful to early treat or to early diagnose the occurrence of diabetes and metabolic syndrome.
I hope they are useful for cancer. By the way, Dr. Kopchik just presented in October, a beautiful lecture, and he had the kindness of showing my work as the reason why he has developed some new drugs that he's testing right now in mice in preclinical studies. And what he's doing, he developed, he's the inventor of pegvisomant, the pegylated form of growth hormone to treat acromegaly, intractable acromegaly anyways. And he has developed compound G. Compound G is another form of pegylated growth hormone. And this pegylated growth hormone given together with standard chemotherapy, it can decrease and it can work beautifully in liver cancer. Let me see if I can get liver cancer, pancreatic cancer, and cholangiocarcinoma. And he has tested these drugs, and he's publishing partially this new compound. So these observations that the growth hormone receptor subjects have diminished incidence of cancer has already served to give them a growth hormone receptor blocker plus the standard chemotherapy. So that is a nice thing. Now, regarding to diabetes, to me, the most important thing is avoid obesity by any means, because obesity is the main risk. Meta-analysis since the year 2017, every single meta-analysis becomes with obesity as a major risk for cancer. Almost every single type of cancers, number one. Number two, in the case of the Laron syndrome subjects, you can see that the relatives, they're a little overweight right now, but they develop all sorts of cancers too. But that's partially, that observation could be a little bit difficult to interpret because in this area there's a lot of contaminants. But obesity in the world, obesity is the worst pandemic we have ever had. There's no metabolic syndrome, hypertension, hyperuricemia, hypertriglyceridemia, you name it, they are associated to obesity.
Thank you. For me, it's okay, and I can move back to the prorector.
Thank you very much. Then we move to the last opponent, and we go to Dr. Stratemans. She is an expert in pediatrician and endocrinology, and I'd like to give her also the opportunity to discuss with you.
Thank you, Mr. Prorector. Congratulations on your impressive work.
Thank you very much.
My first question is, in your thesis, you show that the absence of growth hormone signaling may protect against type 2 diabetes and cancer. This raises the question of whether parts of this pathway could one day be used as therapeutic targets to lower the risk of cancer and diabetes too in the general population. Considering this possibility, what do you think are the most important ethical and medical issues when trying to modify growth hormone activity in people who do not carry a genetic mutation in this pathway?
Okay, that's very important. I would say that if you go from the pathway from the same growth hormone receptor, you know, once insulin binds to the first part of the growth hormone receptor, then the second part comes in, then JAKs get activated, then STAT5 get activated. All right, so then IGF-1, but IGF-1 goes directly and activates the same molecules that the insulin pathways have, and the same molecules that are present after the binding to IGF-1 or insulin to the hybrid receptors. In other words, then you will have the two branches, the PI3K that promotes AKT in one side, and the other side, the RAS-MAP kinases. So these two branches can be activated. Just to give you an example, let's suppose that you have hyperinsulinemia. Hyperinsulinemia either binding to the IGF-1 receptor, to the insulin receptor, or to the hybrids. This can activate, you know, any part of the pro-oncogenes like RAF, ERK, ELK, every single thing can be activated, and every one of those places can be used as a target. Now, that's the mechanistic part of it, but then you have the ethical part. What do we block? And blocking the growth hormone receptor makes sense if you're an adult, you don't use much the growth hormone. You don't use it much, but if you block it, the growth hormone receptor in an adult, to treat cancer, that is justifiable. To do in obesity, the only thing you have to change is diet. So that the ethical part to me really does not exist. Because when you're dealing with cancer, blocking anything, and especially you block just the growth hormone receptor in an adult with an incurable cancer, liver cancer, pancreatic cancer, cholangiocarcinoma, very aggressive form, melanoma, where the viral effect takes such a major role, on mTOR. In those cases, you are blocking. So there's no ethical issue there. But in the case of obesity, what I would recommend is the most difficult part, of course, dieting.
Okay, thank you. And my second question, you already talked a little bit about it, but to what extent could the differences in birth weight between the Ecuadorian-Laron syndrome, which have a normal birth weight, and the Guevara-Roseblum syndrome, which have an intrauterine growth retardation, contribute to your later divergent metabolic phenotypes?
I'm sorry, the last part, can you repeat the last part a little bit slower?
Okay, so in Ecuadorian-Laron syndrome patients, they have a normal birth weight.
Yeah.
And the Guevara-Roseblum syndrome, they have a small for gestational age, intrauterine growth retardation. And how can that contribute to the later divergent metabolic phenotypes?
Well, I would say that that's different. In the first place, I already, when my dear opponent asked me a similar question, I already said this happened and why it might happen, hypothetically speaking. But to me, what produces the effects is the different growth hormone counter-regulation. If you remember, I showed two pictures in which I did 2 hours OGTTs, and just in Laron subjects, and with very little insulin, they get a very normal response. So that is telling you that low insulin is associated with less events of every sort, including obesity, except in the Laron's obesity, the metabolic syndrome, etc., etc. But in the other case, they have very diminished insulin, but they have high growth hormone counter-regulation. Therefore, they develop dramatic changes. So in this case, it's not really the Barke hypothesis. It's different. It's just natural phenomena that occur are completely independent, because what is mutated in the Laron syndrome? Just the growth hormone receptor. In the case of the CDKN1C, there are 3 mutations that can cause this GRS, but this inhibitor acts everywhere. Not only that, but
Dear Mr. Candidate, you are allowed to finish your answer, but you also can decide to stop here. It's your decision.
okay, I can continue and think about it.
Okay.
I will take a couple of minutes. Is that okay?
Yes?
Dear
Yes?
Prorector? So the CDK1 is widespread. For example, let me just give you one example. It acts in the prechondrocytes to chondrocytes. So that's why they miss the growth spurt at puberty time. So that's because the CDK1C acts there too, and I think in the pancreas and in other places. So it's a kind of different mutations. The site in one case is generalized from the standpoint of IGF-1. In the other cases, it's because of local effects in several specific tissues, adrenal, some bones, and they are discrete places.
Thank you very much. I give back the word to the pro-rector.
Thank you.
Thank you very much.
I think.
Dear Mr. Candidate, the time appointed for defending your thesis has now officially passed.
Oh God, thanks.
The degree committee will now withdraw, and we're going to discuss the way you have defended your thesis. The quality of your thesis has already been approved by your assessment committee. And I request that you and your company over here wait the results of our deliberations and the return to the aula.
Thank you very much. Dear Prorector, thank you.
Dear Mr., still call you dear Mr. Candidate, dear Mr. Guevara, Aguirre, the degree committee here present has assessed the quality of your thesis, but in particular we have discussed in depth the way you have defended your thesis, and we also discussed your presentation. In view of its positive verdict and taking into account your previous qualifications, the degree committee here present has decided to grant you officially the degree of doctor. Professor Gavilanes is authorized to confer upon you this academic distinction in accordance to Dutch university customs, and therefore I invite now your supervisor to take the floor officially.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Jaime Guevara Aguirre, the degree of a doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affix it with the official seal of the university.
Thank you, sir. Thank you.
I give the word to Dr. Gavilanes to present your small laudatio.
Thank you, Mr. Prorector. First and foremost, I want to congratulate you on the outstanding way in which you defended your thesis and on the exceptional quality of your work, which has been awarded a doctoral degree at our university. Rather than summarizing your thesis, which would require for more time that I have today, that I get from Mr. Prorector, I would like to highlight a few aspects that make your contribution truly remarkable. Your scientific work and the breadth of your projects reflect a deep and sustained commitment to generate new knowledge in the fields of endocrinology and metabolic disease. You have established a highly successful research line, and your clinical investigations are exerting a significant international impact. What impresses me most is your extraordinary dedication. Throughout the development of this project, you have smoothly balanced your clinical responsibilities and your educational commitments, a rare and inspiring achievement. The work you have defended today also lays a solid foundation for future collaboration between your institution in Quito, Ecuador, the Universidad San Francisco de Quito, and our university. Jaime, you continue to exemplify what it means to be a physician, a researcher, and a leader. This PhD does not mark the beginning of your career. It recognizes a lifetime of remarkable contributions, and I'm certain it will open the way to new and meaningful contributions in the years to come. It has been a true honor and privilege to be part of your supervision team. Working closely with you has been deeply inspiring. Your professionalism is a personal example I hope to follow. Finally, I want to extend my congratulations to your wife, Sandra, and to your children, whose support and encouragement have been essential in this journey.
Thank you.
I give the word back to Mr. Prorector.
Dear, I would call you now officially, dear Dr. Guevara Aguirre. Also on behalf of Maastricht University, our sincere congratulations. And of course, my congratulations also going to your wife, who joins you all the way from Quito. It's a long way, and we are very pleased that you defended your thesis in a very good way. I must say that the team came to a unanimous positive decision regarding your work. I'm also personally impressed, and I already made some notes myself that I would be interested in the future in the brains of those patients to look to this in pathology. Anyway, this is a personal matter. And my congratulations also goes to your promotion team. Only Dr. Gavilanes is present here today, but we noticed that he guided you in the last 7-8 years, that you really could defend your thesis here in Maastricht today, and we are very pleased about that. In my congratulations, no, in my gratitude, it's also to the promotion team, not to the promotion team, but to the members of the Corona. And in particular, I would like to remember to thank both Dr. Kropczyk. They are most likely still there. I'm not sure. Yes, and Dr. Casidor. Thank you that you stayed up middle in the night to ask some questions. And I do hope there was a lot of coffee available to keep you awake in these hours. But thank you very much again. I also would like to thank the other members of the Corona. I'm always saying it takes 7 minutes to ask questions, but it takes more 7 minutes to prepare the questions, so they have to read your extensive thesis. I also would like to thank members who were present here. Most of them are most likely online available, and I think it's time now to celebrate a little bit further. And by that, I'm going to officially close the ceremony using my hammer, as you can see over here, and this session is now closed. How do we proceed? We're going to make a picture here in the aula, together with the members who are online, and later we're going to make a picture outside on the stairs of the university. We will show you, and there is also then some opportunity to congratulate the new, I wouldn't say young doctor, the new old doctor, but there is opportunity for this. Thank you very much. Good.